Okay, so uh, let's focus on massively parallel reporter assays in this lecture, MPRA. So this MPRA lets us measure the functional effects of thousands to millions of sequence variants. So each sequence is linked to a barcode, and then eventually sequencing readouts quantify how strongly that sequence drives things like RNA abundance, decay, for example. So overall, it converts sequence variation into quantitative functional labels. Um, so MPRA matters because it fundamentally changes how we study RNA sequence function relationships. Using this framework, we can systematically test how regulatory elements influence expression. So we can measure 5' UTR effects, coding region features, 3' UTR motifs, how, these all elements, regulate stability, decay, as some examples. And the important thing is, so MPRA lets us study combinations of the sequence features. So that makes it possible to map how multiple regulatory components interact to shape overall expression. So because of this, scale and quantitative structure, MPRA provides dominant experimental data used to train modern predictive models of mRNA function. So let's start with the workflow and then actually start with library design. So each construct contains a short sequence variant you want to test, typically around 100 to 200 nucleotides. So these inserts might include regulatory elements from 5' UTR, or maybe targeted changes that alter RNA's secondary structure, or maybe motifs from the 3' UTR. So then each sequence variant is then linked to a barcode, and actually multiple distinct barcodes usually. So this barcode acts as an independent reporter of that variant's activity. So when sequencing readouts are collected, measurements from multiple barcodes can be averaged, right? So this actually provides technical replication and helps estimate measurement noise. So from here, everything downstream, that is transfection and then eventual expression, sequencing, all those things, they are constrained by what was encoded in this library. Step two of the MPRA workflow is synthesis, cloning, and barcode assignment. At this stage, the designed sequence library becomes a physical DNA library. Each variant must be uniquely linked to one or more barcodes, so later sequencing reads can be traced back to specific variants. There are two common implementation strategies. The first approach, the variant and barcode, are synthesized together on the same oligonucleotide. So the entire fragment is cloned into the reporter plasmid. So here, because the barcode variant relationship is defined, no additional mapping is required. In the second approach, the reporter backbone contains a pool of barcodes. Variants are inserted separately. So this approach is necessary when testing longer sequences that cannot fit within a single synthetic oligo. Both strategies produce essentially the same functional outcome, a physical reporter library, uh, and a barcode linking each to a specific variant. So from a data perspective, this lookup table is critical. Every barcode acts as an indirect pointer to a variant. So if the mapping is incorrect or incomplete, that error propagates into the labels used for modeling. Most designs use multiple barcodes per variant, maybe something like 10 to 20 to average out technical noise and improve measurement precision. By the end of this step, the reporter library is fully constructed, and traceable, and ready for transfection. So once the reporter library is constructed, it is introduced into an expression system. This could be a cell-free extract, cultured cells, or in some cases, even an in vivo model. Each construct produces a reporter transcript that contains a barcode identifying the sequence variant. After expression, we sequence the RNA and count how many times each barcode appears. So importantly, these counts are interpreted relative to the number of DNA molecules originally introduced. So this RNA to DNA normalization is what lets us measure variant effects independent of differences in the library representation. RNA abundance actually reflects steady state RNA levels, and which are determined by multiple processes acting together, such as things like construct transcription, RNA processing, its stability, and how quickly it is degraded. RNA structure can influence accessibility to decay enzymes or ribosomes. And then again, cellular context such as stress state, metabolic condition. So these things shape RNA turnover. So MPRA measures the combined outcome of transcription, processing, and decay. And it is also important to distinguish RNA abundance from protein production. MPRA primarily measures steady state RNA levels. Translation is only captured indirectly. That is to the extent that translation affects RNA stability through mechanisms like ribosome dependent decay. So for modeling folks, the RNA count is the label, right? But biologically, it's a snapshot of the equilibrium between how fast RNA is being made and how fast it's being cleared. So the model is seeing the net result of several overlapping processes. So once the library is introduced into an expression system, so the biological context determines what aspects of RNA behavior you can actually measure. Different systems capture different layers of regulation. Cell-free systems provide, yes, highest throughput. They operate in a simplified biochemical environment, and as a result, RNA output may appear elevated compared to living cells. So these systems are excellent for large-scale screening of intrinsic sequence effects, but they don't really capture full cellular regulation. So whereas cell-based systems introduce a more realistic environment. So here RNAs experience, decay pathways, quality control, for example. And then we know that cells can detect, foreign RNA and activate innate immune and stress responses. And actually, this differs dramatically across cell lines. So here throughput is lower, but the measured RNA dynamics better reflect what happens inside real cells. And then when it comes to in vivo MPRA, it extends this realism much further. Reporter libraries are delivered into living organisms, and then measurements in happen, and they actually reflect a true tissue context. So here we can capture cell type specificity, systemic signaling, immune response, and overall organism level regulation. But here, throughput is much lower, right? But biological relevance is highest. So what we need to understand is that these systems are not interchangeable. So they form, kind of a hierarchy. Cell-free gives you scale. Cell-based tells you about mechanisms a bit more, and then in vivo provides reality. So practical MPRA pipelines, obviously need to use all three. So after the library is expressed, we quantify the activity of each sequence by sequencing both the DNA and RNA. So DNA sequencing tells us how many copies of each construct were delivered, whereas RNA sequencing tells us how much transcript was produced. So, and again, this transcript, it represents steady state abundance, like we discussed. So anyway, we then compute the RNA to DNA ratio for each variant. So this normalizes RNA output by how many copies of the construct were present, allowing us to compare functional activity across sequences. Importantly, this ratio doesn't isolate individual mechanisms like transcription rate or decay rate, but um, it reflects actually their combined effect. So we just learned that um, taking the ratio of RNA to DNA estimates activity related to input, but uh, raw ratios are messy, so they are highly skewed. A few strongly expressed variants can dominate the scale. So we take the log of the ratio. The log transform can do two important things. First, it makes fold changes symmetric. So a twofold increase and a twofold decrease are equally spaced. Second, it compresses extreme values so highly expressed variants don't overwhelm the dynamic range. In practice, we compute log 2 of RNA plus 1 divided by DNA plus 1. So this plus 1 prevents undefined values when counts are 0. So it's a practical solution, but it does influence behavior at very low counts, which is why filtering and coverage checks still matter. We know that standard MPRA measures RNA abundance, not translation directly. And there are several specialized MPRA formats. They let us probe translation. One approach is ribosome profiling of reporter libraries. So here we sequence ribosome protected fragments from each variant. So this tells us where ribosomes are bound and how densely they occupy the transcript. It provides mechanistic insight into ribosome recruitment, scanning, and pausing along the mRNA. A second approach measures protein output directly using a reporter readouts like fluorescence or luminescence. And then again, a third strategy uses cell-free translation systems. So these isolate sequence intrinsic translation behavior by reducing cellular regulatory complexity, letting us probe features like initiation efficiency and local RNA structure effects. So important thing here is these assays measure different layers of gene expression cascade. So they generate different types of labels and answer different questions. Stability MPRA is designed to measure how quickly different RNA variants are degraded over time. Instead of sampling RNA at a single time point, we measure abundance at multiple time points after expression begins or after transcription is halted, and track how each variant declines. So this lets us estimate apparent decay kinetics for each sequence. So these measurements capture sequence features that control RNA turnover. For example, poly tail shortening often precedes degradation. So sequences that accelerate deadenylation tend to have shorter half-lives. UTR motifs can recruit decay factors or protective proteins. RNA structure can expose or shield degradation sites, and Interactions with ribosomes can influence translation coupled decay. So an important thing is, the stability MPRA focuses on the disappearance of RNA, not production. So it attempts to isolate the decay component of RNA abundance, basically. Biological interpretation comes from comparing how quickly different variants decline relative to one another after normalization for initial levels transcription rate. So stability MPRA provides a direct experimental handle on sequence encoded RNA turnover. But there is one important caveat for our training data. When we measure stability, we are trying to calculate decay rates, but we usually have to shut down the cell's natural transcription to do it. So this puts the cell under stress. So the model might actually be learning how RNA is cleared during a stress response rather than a normal physiological state. We discussed about barcodes in previous slides. So each barcode acts as a replicate measurement of how that sequence behaves. But again, barcode measurements are not perfectly identical. Variability can arise at many levels. Biologically, RNA molecules are produced and degraded stochastically. Cells differ in their internal state, plasmid copy number, and expression environment. Even when the sequence is identical, these factors introduce natural variation. And then you have technical process. They also add noise. PCR amplification can preferentially expand some molecules. And this is actually called PCR jackpotting. And then again, sequencing depth, that's also not perfectly uniform. And then maybe a library preparation may shift barcode representation. So because of all these things, individual barcode measurements are noisy observations of an underlying sequence effect. Using multiple barcodes per variant lets us average across this variability. So this reduces random noise and improves the precision of our estimate of how the sequence truly behaves. So to continue from previous slide, not all zeros are the same. In MPRA data, a zero RNA count can mean different things depending on the context. If a variant has high DNA counts but zero RNA, that means it was clearly present in the library, but no RNA was detected. So this can reflect strong instability or extremely low expression. So these zeros may represent real biological effects and should not be automatically removed. On the other hand, if both DNA and RNA counts are near zero, the variant was barely represented in the library. In that case, yeah, there is no reliable baseline, and the zero is, not exactly informing us anything. So before filtering, we'll look at the DNA coverage. And low DNA variants are often excluded. High DNA, zero RNA variants are usually retained. So the key idea is, that is zeros are not inherently noise. Their meaning depends on the context. And when it comes to synthesis bias, so MPRA libraries begin with array synthesized DNA oligopools. This synthesis process is not uniform. Certain sequence features make some variants easy to manufacture, clone, and amplify than others. For example, GC-rich sequences are often underrepresented because they can form stable structures that make synthesis a bit harder. And then homopolymers and repetitive regions frequently fail during synthesis. Error rates also increase toward specific positions along the oligo. After synthesis, additional biases occur during cloning and amplification. Some variants enter the plasmid library more efficiently. And then again, barcode assignment can also introduce mismatches between sequence and measurement. So all of these things happen before any biological experiment actually. So as a result, the MPRA library is not a neutral sampling of sequence space. It's already shaped by chemical and technical constraints. This means the data distribution used for model training reflects both the sequence biology and library construction bias. So for modeling, this is critical, right? Because observed counts reflect what was successfully manufactured and delivered, not just what is biologically functional. So most of these biases actually arise from synthesis and handling limitations, not necessarily biological regulation. When it comes to assay artifacts, we know that every assay system introduces its own artifacts. In cell-free MPRA systems, RNA operates in a simplified biochemical environment. Uncapped 5' triphosphate RNA often shows reduced translation initiation and innate immune sensing is largely absent compared to living cells. So there is no cellular compartmentalization or regulatory feedback. So because of these things, these conditions can inflate apparent expression and reduce sequence selective clearance. So, and then we know that the cell-free systems are powerful at when it comes to scale, but they simplify regulation, right? So now, when it comes to cell-based MPRA, we have opposite trade-offs. So the biology is more realistic, but also more variable. Transfection efficiency differs between cells. Plasmid copy number varies. Cell health, stress pathways, confluence, and passage number all influence RNA production, degradation, and translation. A translation coupled decay can selectively destabilize specific sequence features. So cell-based measurements capture, bit real regulatory biology, but they also introduce substantial biological noise. So here is what's critical for therapeutic mRNA modeling. I mean, just if we take one example, many cell-free systems use uncapped RNA, but we know that therapeutic mRNAs are capped. So that means, the assay bypasses cap binding machinery entirely. So if a model relies too heavily on this particular kind of data, cell-free data in this case, it may underestimate the importance of 5' UTR features that are essential for capped mRNA translation in living cells. So the basic point what we want to stress is one should understand the assay context, and then clearly understand the biology. Then you would know what your model is actually learning. Batch effects are systematic differences between experimental runs that change how the same sequence behaves. Importantly, batch effects are not just technical noise. They arise from a combination of biological state differences and experimental handling. Cell passage number shifts, regulatory programs over time. Metabolic and growth states influence transcription, translation, RNA turnover. Transfection efficiency changes how many plasmids actually enter each cell and how they are distributed. So, I mean, even media composition or reagent lots can shift as stress signaling and cellular physiology. In these MPRA experiments, library composition itself can differ slightly between preparations, which also changes which also changes these ratios. So the key point is this: batch effects produce systematic shifts, not random fluctuations. The same sequence can produce different expression levels simply because the cellular environment has changed. So this is why replicate structure, normalization, and careful experimental design are essential for interpreting data reliably. And so what matters for ML training is batch effects come from real biological state changes, cell stress, passage number, these kinds of things, not just technical variability. So if uncorrected, the model learns batch, not biology. So this is why cross-batch normalization and batch correction are important. Many MPRA libraries are built from parent sequences with small changes. Variants derived from the same parent often share similar activity patterns. If we randomly split these nearly identical sequences into training and test sets, The model can memorize subtle sequence features and appear highly accurate. But that performance is, I guess, bit misleading. The model is not learning general rules, it's recognizing close relatives. To properly evaluate generalization, we should group related sequences together and split data by sequence family. That way, the test set truly contains unseen sequence backgrounds. So this prevents inflated accuracy and gives a more honest assessment of model performance. Let's summarize. And then we already understood that MPRA doesn't directly measure intrinsic sequence activity. It measures RNA abundance produced by each construct under specific biological conditions. So that signal reflects multiple processes acting together, transcription, RNA processing, stability, translation coupled decay. And again, all these are modulated by cellular state and the experimental system you choose. Um, and then we discussed that the expression system matters a lot. Cell-free, cell-based, in vivo setups each capture different mechanisms. Barcode replication helps us average out stochastic variability, but technical biases from library synthesis, assay artifacts, and batch-to-batch biological differences still shape the data. So the core takeaway can be that the observed expression is not just a property of the sequence. It emerges from the interaction between sequence, biological context, and measurement system.